Hello, everyone, and welcome to today's broadcast, ONETest PathoGenome, a target hybridization-based next-generation sequencing assay to tackle undiagnosed infections. It is presented by Mohammad Qadir, PhD, the President and Chief Scientific Officer at FUSION GENOMICS. I am Judy O'Rourke of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Qadir. I will now turn the presentation over to him.
Hello, everybody, and welcome. First of all, I would like to begin with thanking the organizers for according me this opportunity, and to all the viewers and viewers as well, welcome. So we are here to talk about massively multiplex diagnostics for infectious diseases. The question is that we would want to address is, what do we mean by massively multiplex, and why do we need massively multiplex diagnostics to begin with? So before we go any further, just an upfront conflict of interest. I am the Chief Scientific Officer at FUSION, as mentioned, and thus do have financial holdings in the company. Also would like to mention that the assay and the data, which I am sharing with you today, most of it is unpublished, and also the assay at this moment is for research use only, and thus it is not for clinical or diagnostic purposes for any human or animal testing. And if I'm talking about other companies and their products or anything, they are only for educational purposes on an as-is without warranty basis. So without further ado, let's begin, and let's start the answer. Or answer the question, why do we need massively multiplex diagnostics? As you can see on your screen, the problem in infectious diseases, and the esteemed audience will also be aware of it, that many of the hospitalized patients, so we are restricting our discussion from here onwards to patients who are hospitalized, and during their stay in the hospitals, many patients remain undiagnosed. For example, you have the examples ranging from pneumonia to diarrhea to meningitis and sepsis. Even what's not on the screen, an area where the current molecular diagnostics have made a huge leap, which is the PCR-based panels for upper respiratory syndromes. I was certainly surprised to learn that, but many of you may already be aware that during peak flu season, for example, where you have patients in the ER who are clearly in distress because of upper respiratory or respiratory involvement, and yet when you run them on these PCR panels, the assay still comes out as negative. So, and I'm sure all of you are already aware of the cases in sepsis and GI. So this is the problem. Now, the other thing about this problem is it is a medical problem, and at the same time, it also creates a massive burden on the healthcare system. And that burden can be viewed as, for example, by conservative estimates, by others, published data and such. The cost of the cost of a patient remaining undiagnosed because of idiopathic or again undiagnosed infections throughout their stay in a hospital or during their stay in the hospital, the yearly cost in the United States alone is approximately $27 billion. Now, it is a big cost, and the breakups are also presented for some of the things on the side. If this was only the thing, we'll say, okay, it's one thing. This is a direct cost. But what is also very medically also interesting about this, that these patients also are part of a major contributor to then indirect costing, which results in things like antimicrobial resistance. So how do we solve this problem? Well, as smart people say, that in order to address a problem, one has to understand it. And the way we made it us understand it, or the way we view this problem as an iceberg problem. And what you see on your screen is a cartoon iceberg representation of how we feel this problem has remained and will remain until we address the magnitude. What this thing shows is that if you look at the tip of the iceberg, it represents your FDA-approved molecular diagnostic assays for infectious agents. According to latest estimates, the 2008 publications as well, there are about 1,415 known pathogens, and the FDA-approved assay covers only about 16% of them. And then underneath the surface, you also have your antimicrobial resistance, which again, which actually adds an additional $26 billion annually to the economy in costs. And that is just US alone. And then you have your undiagnosed infections. And then underneath those infections, you have your pandemics, where we hear about a Zika outbreak and things like this. They are all part of this same iceberg. And then underneath them, you have your emerging and re-emerging pathogens. For example, pathogens can be in your population for a long time before they emerge and become a pandemic. And underneath all of that is your zoonotic reservoir, which are the pathogens which exist in the animal world around us, in the environment around us. And then when they jump from the animal world into the human world, then we have major problems. So we view infectious disease not just that patient who's remaining undiagnosed, but what is needed is to address this iceberg, this hidden nature of the problem. And so now we ask another question. Can we address this entire iceberg? And the answer is actually surprisingly yes. And that comes from the power of genomics, especially we are in the post-genomic era. And the way it is addressed, or the solution, is a form of whole genome sequencing called as whole metagenome sequencing, in which what you do is you take any biological sample or any sample, and you would sequence all the DNA or all the RNA in that sample. Basically, you'll have a soup. You can take blood, sputum, nasal wash, biopsies. You can take environmental samples. Then you would prepare all the DNA or RNA in it, and then you will put it onto a next generation sequencer. And obviously, at the end of that, you will end up with your sequence material. Now, what happens with the next generation with whole metagenome sequencing is that when it gives you your results, you have everything in it. You have your normal human DNA in it. You have your normal bacterial DNA in it. You have your normal viral DNA, and then also your pathogen DNA. So it gives you all this data, whereas what you are here is your pathogen DNA. And for this, on these purposes, the diagnostics and precision medicine are using whole metagenome sequencing. It is not an ideal solution to be used to address the problem at large. Why? Because it has a high cost. Yes, we understand if a median cost of a hospitalized patient can be in thousands of dollars, you can spend a couple of $1,000s to diagnose them. But then there are added problems, which is that it is still difficult to use. One of the reasons, as I just mentioned, why it is difficult to use is because of all the data that it will generate, and then you have to sniff through all that data to find the pathogen in question. And it still takes it still takes longer to perform, and which is sometimes outside the boundaries of what is required in a clinical setting. So at this point, we ask the question, okay, I mentioned that we have an answer, but then I'm also saying we don't really have an answer because it's not that practical. So where we should go from there. So here we talk about, which is also a variation in molecular biology, which is known as target capture. In target capture, what you do is you start with your same biological sample, but then what you want to do is enrich the things you're looking for. A PCR or PCR panels are actually an example of target capture because you go in and amplify what you're specifically interested in. The human exome sequencing is also example of target capture. The difference is that PCR panel is based on 2 PCR primers amplifying, whereas probe-based hybridization, you make complementary DNA or closely related DNA to put into that soup, if you may want to call it, and capture the DNA or RNA of target of interest. And once you have captured it, you will sequence it. And the idea is that after you have done your target enrichment or target capture, once you sequence your sample, you will end up with what you were looking for. Either it is there or it is not there in a binary sense. And it also becomes absolute because you are sequencing, you're obtaining directly genomic sequence of whatever you are trying to capture. Now, before I go any further, I thought it might also help the audience, and I'm pretty sure that most of you are already aware of it. So just a refresher is that a bit of the history of target capture. The target capture or a complementary DNA hybridization, it was first discovered by Dr. Edwin Sutherland in 1975. And Dr. Sutherland actually went and won a Nobel Prize for the discovery. And this was the underlying technology for all the southern blots and northern blots and western blots. Then the next major iteration of the technology came in the form of DNA microarrays, started in around the 90s by Dr. Patrick Brown. And then the third iteration came with the human exome in 2009. Also, what changed in these iterations is that in the time of Dr. Sutherland, or when you're doing a southern-northern blots, your target, whatever you are interrogating, it actually is immobilized on an immobilized surface, which used to be a nylon membrane and such. And then you will add your probe or your complementary oligonucleotide to go and bind it. When it moved from that into DNA microarrays, what happened is that nitrocellulose or a nylon membrane was then replaced by probe with your oligonucleotides, which were then put on a glass slide. Then you would add your interrogating DNA or RNA onto it and see if it would bind. You would have a fluorescent signal. And in case of human exome, it went a step further, where what it is called as a liquid phase hybridization, and hence the name liquid phase capture, in which both your target and your probe are both in a liquid. What changed over here is that what you do is when you have your complementary DNA, you attach a streptavidin molecule onto it. So once it attaches to the thing that it wants to bind to, its target, and then you can put that tube on a magnet, and that way you can take it out of the solution. So to give a final overview of this, the way target capture works in the liquid phase hybridization, that you have a piece of DNA, and you take that piece of DNA, you have a streptavidin on it, and you know what that piece is, and you use that piece as an enricher for whatever is in the solution. If the solution contains what you are looking for, this piece will go and bind with that piece as shown in the cartoon, and then you can pull it out of a solution using a magnet. And this is exactly what Fusion is also doing and what the ONETest PathoGenome is. Over here, I like to add that we are still a very pre-commercial, so we are a very much an R&D enterprise, and hopefully you will see that we'll be sharing a lot of data with you, and hopefully I will not be trying to sell you anything. We want this to be a scientific presentation. Anyways, but we still have to talk about the underlying technology to effectively communicate to the audience. So we have invented this is what we call it, or others call it, as a disruptive DNA RNA capture technology. And what it allows us to do, it makes these oligonucleotide probes, which we can direct towards all infections, and that could include virus, bacteria, fungi, and parasites. It has been optimized to work with all biological sample types: blood, sputum, stools, swabs, and others. It's a single tube assay, which means that you add your sample, I need to correct that, you add your nucleic acid. So whatever biological sample you have, you will then isolate your DNA or RNA, depending upon what you're interrogating, or both, and then you would add it into the tube. And once your target is enriched, it's put onto the sequencer, and then all the data goes to a cloud where it is analyzed, and you are provided with a report. And the most intriguing thing about it, that when technology will be commercially introduced, it will have approximately a 24-hour turnaround time, which means that from DNA or RNA to your result, it will provide it within 24 hours, which we want to call it as a clinically relevant time frame. One more thing about it, and then we'll move into the data. It is a kit-based sample to result assay, which means it's not a service. It's a box which has everything you need. It has a very simplified laboratory workflow. It comes with its own suite of analysis, which is cloud-based, and it is a completely secure and compliant cloud solution, which is using IBM as its at its backend. One more, what I want to add over here is, and the platform or the assay has actually already been tested in some of the most diverse conditions. So, for example, it is under clinical studies at the European Union, part of their genomic effort on their sentinel patients. This is the flu assay, or the flu version of the assay, and it can capture and identify every known flu. And it also provides you the complete genomic sequence of the HA and the NA regions. It is the same assay, and now, as I mentioned, that it is the capture technology which is disruptive. So what is special about that capture technology is that the way our probes are designed, they are not designed against any specific species or a reference genome. They are designed against all the known evolutionary lineages across all known species that we have information for. So for example, what does that mean? That when we have an assay for flu, it is regardless, it would work on humans, it would work on birds, and it would work on swine or pigs, or for that matter, any other organism, because the probes are derived to capture evolutionary lineage. One way of thinking of it is a way that if you take one of our probes and you blast it on NCBI, it will not have an 190, 80, 70% match to any one single sequence, but it has enough matches against an evolutionary lineage that it can go and capture and bind to the target, and you are able to pull it out. And the efficacy of it is that, for example, the second example on your screen is about a bird flu version of the assay, in which what you see in that tube is actually soil and mud, which is collected around the rivers, and it is in the path of the migratory birds. And the migratory birds are dropping their bird droppings, and those dropping is in the mud along with all the animal fecal matter and everything else and the plant matter in it. And by using the ONETest platform, we are able to capture the flu, the avian flu genome from this and be able to sequence it. Similarly, we have a project with the Sunnybrook Research Institute in Toronto. It is recently also funded by Genome Canada and Ontario Genomics Institute, where what they have done is put these air canisters around the pig farms. And using the assay, you can process the air in the pig farms. And yes, we can actually capture pig flu virus from those air canisters and give you the genome of the pig flu, which is then subsequently sequenced. So this is how this tells you about the specificity and the sensitivity of the platform. Now we'll go into some of the data, which is more human-centric. Here are some hepatitis C patients, and these are very unusual patients. These were among the most hard to diagnose patients that you can ever find from a genotyping perspective. What it shows is that you have we don't really call them patients. These are this is the RNA samples, which has the RNA belonging to hepatitis C. Now, if you look at it and you focus on the green dot, which is labeled as 1H, what you see there is that this is an infection. This is a hepatitis C infection, which has mutated about 16% for its E1 gene and about excess of 13% for its NS5B gene from every known hepatitis C that was at that time available. So the blue dot of the zero axis is every known genotype of hepatitis C, and this one patient or this hepatitis C genome has mutated excess of 16%. And the reason it's very important is that we know that our assay has an evolutionary distance capability of capturing anything which is about 20% divergent. Just to give you an example of a 20% divergent, if you look at a gene, a slightly conserved gene, like the globins or things like these, a 20% divergent would mean is the genetic distance for their gene between human and Drosophila actually is smaller than 20%. So just gives you an idea that the pros, because they were designed against the evolutionary lineages, they were able to go and capture the hepatitis C genome, which had mutated to such an extent. And all the samples in this study were things like these. There were also even more interesting samples. So if you look at the table on your extreme right hand, and if you look at the sample number 7 and sample number 8, we were even able to determine that that this sample had 2 hepatitis C distinct infections present, one belonging to the genotype 2B, the other to 3A. The sample 8 had 3A and 1A, and because it is an RNA sequencing based assay, the very nature that you are capturing everything from a solution and you are sequencing it, it makes it absolute quantitative. So from that sense, we were able to determine not only the proportions of these 2 genotypes present in each of these samples, but we were also able to determine the viral loads in IU per ml. The graph in the middle just gives you a depiction that how during a treatment, if you have a person going through treatment, you can then track if their genotype of the infection being targeted, how it is changing, or is it being eliminated. The next slide is about the sensitivity of the platform. These are about 50 influenza clinical samples from the Sentinel screening program, and they were also then tested on PCR in a clinical laboratory, and by interpolating and then they were also tested on the ONETest. And what it shows you that the ONETest has a direct comparative sensitivity from the PCR world to about a CT of 53 and 54. Now, as you know, people, especially who come from the PCR world, that a straight up CT of 53 or 54 does not exist, which is true. But what it means, it is comparative to a nested PCR at about when a sample is sequenced at about 1.5 to 2 million reads per sample. Moving forward, as I mentioned, because we are capturing the entire genome or regions of the genome, here is the example from hepatitis C, where what you see on your screen, those gray things, is actually the entire genomic or the gene was captured and sequenced for the E1 and NS5B region. We routinely achieve a depth of about 300, and in this example, this was a very uncommon 3A genotype. The next example is actually a novel 3B. So what it means is that your strain doesn't have to be known, because you're capturing the entire genomic region and you are sequencing that genomic region. You can also detect novel things. Moving forward. Okay, so far the data we discussed was largely coming from single organism studies, whereas the intent of the assay is to be massively multiplexed. Now, you would agree with me that yes, we are designing the assay against all known pathogens, but it's virtually impossible to test them against all known pathogens. So the approach we have taken here at Fusion Genomics is to test them against both clinically challenging situations as well as scientifically challenging situations. So, for example, an upper respiratory, even though we know that there are a lot of PCR panels, it's also a technically challenging situation, especially during peak flu seasons, and especially when the PCR test gives you a negative. And then we have also tested it in scientific challenging situations, for example, in skin ulcers, where it is very difficult either to culture those bacteria or even if you can culture them, is to differentiate various types and subtypes. Similarly, we are undertaking studies in transplant patients for various things, also in GI disorders and also encephalitis meningitis. These are just some of the studies we have undertaken today. I'll be sharing some of the data from the upper respiratory panel and the skin ulcers. Unfortunately, we cannot share the manuscript because it is under preparation, and hopefully it should be coming out, and you guys will be able to then read it in the manuscript. And also because a lot of work and effort has also been put in by our academic partners. So we believe that they should be really who should be presenting it and not us. But we'll still talk about the data or the take-home message from these studies, that what does it mean when you take a genomic assay and you pit it against whatever is out there. Just a little overview that if you were to just look at the upper respiratory, how did we design the assay? So on your left-hand side are all the pathogens out of the pathogenome, which are representative of the upper respiratory pathogens. Then you can also see on the screen all the types and the subtypes which are being targeted. And we are targeting not a single gene or a single region. We target, in most cases, multiple regions on the genome. And that is determined actually in exclusivity by what is already known about the regions, one, and two, how clinically important information in that is available in those regions. So for example, in case of flu, we would target and capture 100% of the HA and NA. And as you can see, that ranges anywhere, in the case of metapneumo, we target about 98% of the F protein and about 88% of the nucleoprotein, and similarly. But again, because the assay is a dynamic assay, we can always increase that coverage or add a region if we think any clinical information is available in that region, or as new information become available, we can easily add probes targeting any region of the genome. But just to give you an idea, so what it means when you say that your gene coverage is 100% is that if in a sample you have flu A or flu B and you run this assay, the probes will go and bind the entire length of the flu A and flu B. So when you will enrich and pull it out and you will sequence it, you should be able to reconstruct the entire gene in its entirety. Moving forward, so today I would be talking about, so this is some of the patients, small samples. Again, calling these patients is incorrect. These are just anonymized RNA and DNA, and these were already tested on the Luminex RV FAST platform. And then they were also tested for the ONETest PathoGenome for the RNA and the DNA. And as you can see, we were in almost 100% concordant, except for this sample number 18. And I apologize for the quality of this slide. It happened because of a conversion between PDF and PowerPoint. So my apologies. And what is interesting is that, yes, the sample number 18, which was originally on the Luminex as a Legionella, okay, you can read it. It was identified by our end as a flu A, and we are looking into it. But other than that, we were able to diagnose everything that was originally diagnosed. Now, in addition, for example, what was also interesting to us is, for example, if you look at sample number 5, which was negative on the RV FAST, but on the ONETest DNA, we were able to determine that the sample had DNA from pneumoniae, from S. pneumoniae. Then, if you also go towards the very end of the sheet, you also see trace. What that means is, because the assay is sensitive and the assay is capturing the genomic regions of these viruses, we were also able to, in all these samples, or in most of them, we were also able to recover traces of other pathogens. Now, as I mentioned, that all the data is automatically analyzed. One other thing which is part of the FUSIONCloud is that, based upon all the data which is already existing in the cloud and our machine learning algorithms, it can actually tell you exactly if what you are seeing is a clinically significant infection, or is it just a trace? It's just there. What does it mean? We don't know. As more sensitive assay becomes available, I'm pretty sure we would know about it, but I just wanted to mention that the assay also have this capability. Now, after we saw this, we became, again, we already knew, but it reinforced our understanding about the about testing the negative samples. So in this study, which was conducted with the European Union CDC, one of their institutes, it is a 300-patient study, but we'll just be talking about the negatives. And they were performing, because these are the sentinel samples, they were performing about 198 different PCRs per sample. And what it tells you, that even after that, they had about 47 cases which were negative. Out of those 27 cases, on our end were also negative or concordant. But interestingly, in 20 cases, we found positivity, which was clinically significant. And it happened because of two reasons. One, because it's a genomic assay, we have no restriction of how many pathogens we target. And because we were targeting every known upper respiratory pathogen, or that's part of the panel, we were able to also identify many infections which couldn't be otherwise targeted using PCR assays, or they were already doing 198 assays, so it becomes a problem. And we were able to easily identify COV, HMPV, and PIV. And also, what also happened is that we also were able to capture additional pathogens. Again, the pathogens that either cannot be tested because of the complexity of the assays or the number of reactions, the sheer reaction that you would have to do. Even in previously identified infections, we were able to identify new co-infections which belong to anywhere from the corona to the metapneumo and to parainfluenza and ADV. Moving ahead, what it also shows, and I've alluded to it, that the assay is also fully quantitative, and it gives you a clinical level detection of the sensitivity at only about 2 million reads. What that means is that, remember we talked about the whole metagenome sequencing. Well, when you're doing a whole metagenome sequencing, you really need to do about 10 million genomic reads. So in that, if you remember, we talked about that in a whole metagenome, you take the tube and you sequence everything. Well, if you want to really sequence everything in that tube, all possible DNA, RNA in it, that would still cost in excess of $20,000, $30,000. But what people do is they sequence about anywhere from 10 to 30 million 10 to 30 million reads or sampling of the DNA from that tube. So when you do whole metagenome sequencing, on average, you need to do a minimum of 10 million genomic read samplings from the tube, or normally about 20 to 30 million. In our case, all we need to do is sample 2 million, because remember, we are enriching. And the third point is that 2 million reads, we have about 5 times more sensitivity if you were to take that same sample and do 30 million reads for whole metagenome, which means is that in order to be equivalent in the amount of sensitivity that the assay is giving you, you would need to sequence 5 times 30 would be about something about 150 million reads, which is very close to our estimates also, especially when we're doing the environmental samples, just like in the case of bird flu, because a scoop of soil has about 10 to the power of 6 bacteria in it. So because it is so dense, you need so much read. There's so much sampling. And the graph on the right, what it tells you is that regardless when we sequence a sample on the platform, we can instantly put that sample in amount of its quantity and titers expression into low, medium, and high. So without looking at anything else, the assay instantly can quantify an infection into these categories. And yes, it can provide you your IU, ML, and everything, all of that, but it can also give you a complete overview that based on all the samples have been tested, where does this sample stack up in terms of its quantity that is present. Moving on, here is an example from the skin infections. As I mentioned, that skin infections are very hard. So in this instance, the assay from the pathogenome, the skin infection portion of it, is designed to identify all 63 pathogenic species wherever they are available. And on the right-hand side, it shows you an example of a standard serial dilution, and we're only showing M. marinium, where what it shows you that the assay can go down and detect as little as about 2 CFUs, and also where your 16S specific or even the genetic specific PCR would start to fail, the assay keeps on working. In summary, the platform and the assays so far, because we also sequencing very less amount, it is actually about 10 to 30 times cheaper to use than whole metagenome sequencing from your sequencing perspective. It has the ability to give you 3 to 9 times faster results. It already provides you about 5 times to 50 times the clinical sensitivity of a whole metagenome assay. What it means is that, and we already saw that in the space of the PCR, it is as sensitive at 2 million reads as a nested PCR. And it provides you about 100x CFU, more than 100x CFU detection when compared with a generic 16S PCR. So this is the only slide about the product. We want to launch it in about 2019 as an RUO LDT. It will be priced at $300 and will have a turnaround time of 24 hours. And somewhere down the road, we'll also have, because the reason it has to be RUO, because as I mentioned, you can, you can certainly try, but having an FDA-approved assay for $1,400 is not, I don't know how long it will take. So it will also have an FDA CE Health Canada approved version for specific syndromic conditions like sepsis, GI, and all of that. And that will have a turnaround time, depending upon the syndrome, anywhere from 12 to 24 hours. That will be priced between $300 to $1,500. And sometime down the road, we also plan to introduce a single agent cartridge-based assay. Applications of the platform. I hope I have convinced you that using a genomic target capture approach, we can handle the problem of undiagnosed infections. It can be used in epidemiological investigations, transmission and tracking of hospital-acquired infections, antibiotic resistance, surveillance in hospitals, and environmental monitoring. So what does that lead us if you go back to that iceberg? Here, as I mentioned, that we believe that the only way of addressing the problem is addressing the entire iceberg. And we believe that time has arrived to address the iceberg, and we're trying to actively address the iceberg. And we'll be very happy to hear from you, your questions, your concerns, or anything around it in any way we can help. With that, thank you very much. I really, once again, want to thank the organizers, as well all of you who have who are here listening to this event. I hope it has been worth your time. Thank you very much.
Thank you, Dr. Qadir, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. Let's get started. Our first question is, how can this technology be employed at a hospital?
Oh, well, that's an excellent question. You can employ it at various, you can actually take a staging approach. For example, you can employ it for a single indication where you think that is the most important aspect in your institute. And we have many instances of that. So for example, we're working with a major hospital who had really the problem with the GI infection. So yes, you can bring in the technology as a GI infection, but then where it really helps and shines and becomes very cost effective is that if you convert your molecular testing and even your routine testing, especially culture, onto a genomic platform like this, what it means is now all of a sudden you don't have to maintain all those incubators, all those cultures, all those hundreds of PCR primers. You can have a single assay, which you can then batch at the end of the day and run all your samples on a single assay. What it also does to you, that it actually brings your sequencing costs down. And your assay costs. For example, we have done this analysis where if you were to do, say, only 96 patients or samples every time, your sequencing costs will reduce to about $30. So yes, again, I hope I've answered it, is that yes, you can introduce the technology as to address a specific problem or to adopt it as a platform. And the other advantages of adopting as a platform also, that it also allows you to remove so much excess things that have to be maintained and cultures and things like that.
Thank you. Does the assay need any specialized equipment?
Good, yeah, okay. The assay can be, at the moment, can be ran as an RUO or an LDT in any molecular diagnostic laboratory, and all they need is a medium-sized DNA sequencer. The technology is compatible with all the DNA sequences, because if you think about it, all you're doing is taking that DNA, enriching it, and putting it onto the sequencer. We're not doing anything to the sample. That's also an important feature. The integrity of the sample and its relative ratios and expressions, nothing changes. You just enrich it and put it onto the sequencer. So yes, to answer, you would require a next generation sequencing instrument.
Thank you. And we have time for one more question. How will the assay be reimbursed?
That's an excellent question. So in the hospitalized setting, the assay can be easily reimbursed under the DRG pathway, which is the diagnostic related group. When the assay is actually introduced as an FDA approved or Health Canada approved version for these syndromes, it will follow the current CPT codes for those syndromes if they exist, or we will file new ones if they need to be.
Thank you. I would like to once again thank Dr. Qadir for his presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 13th, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.